Jai Hind everyone, I am Pankaj Kumar and today's lecture is about the DNA replication. First of all, I will tell you the important points which is the continuation, which will be the continuation of the lectures. So in the first lecture, it means this lecture, we are going to discuss about the discovery related to the DNA replication. Then we will see the actual DNA replication in the next video. So in the next video, what we will see? In next video, we will see about the initiation, elongation and termination. So that is the actual part of the video, and this is the base, like foundation of that upcoming videos. After that, we will see the telomerase replication and the disorder related to the DNA replication, and then we will see the practical research problems when you will do a research lab work. Then what type of DNA replication type of problems, or the PCR related problem, or the gel electrophoresis problem, or the what are the actual research problems or research results were showing in the papers, in research articles. That articles we are going to discuss. In the last, we'll solve the questions related to the GATE or ICMR or the CSIR or other type of examination like IIT JAM or GATB. So you probably you will be thinking that it's a like a hotspot, but it's not actually. It's a complete understanding of the DNA replication, which can be helped in the different examination. So stay tuned with me for the videos. And one more thing, that if you want to join a live class, please, there is a link, visit it, given in the description box. For the registration, you have to go and register yourself for the live class. You'll get a demo class, 3 demo classes. If you like the demo content, please subscribe and then buy the course. Now we'll see. So see, the discovery is something in which we will discuss how they found the DNA replication. So Watson and Crick told Watson and Crick, given the DNA structure. Okay. So the few things we have to understand and remember, I will write here, and you also copy. The DNA is a double helix structure. It is a what? Double helix structure. And there are several type of DNA, but we will discuss about the BDNA and ZDNA. So BDNA is more prominent and more known in our text and in our body also, in living world. So BDNA and ZDNA is something like this. So from this pitch to this pitch, this is one pitch. Okay. So this complete pitch is called it's called as a one pitch, and the length of this is around, in BDNA, this is a pitch, and this is a nitrogenous base. This nitrogenous base pair having a twisting. So suppose this is a one nitrogenous base, and this is another nitrogenous. So there is an angle between this. You can see this angle. So this angle is having some value. So in B and Z DNA, that is both is different. And remaining that there is a major and minor group, and this is a width. This is a width, and then angle, and there is a puckering, which are the important thing for the higher studies. In BDNA, the pitch is about 3.4 angstrom, and in this, in ZDNA, it is around more than 3.4, and this width is 20 angstrom. Let me see again. Angle is around 36 degrees approx, or puckering is C2 prime puckering. Okay. So ZDNA, its width is around 22 angstrom, and then so this is a 30. If you are writing in the angstrom, it will be 34 angstrom, and if you are writing in nanometer, then it will be 3.4 nanometer. So I want to write it in angstrom, so I have to write it as a 34 angstrom. And the same thing, if you will see here, it will be around 36 angstrom. Now, the angle between this two nitrogenous base pair, it is around 44 degree. This is C2 Indo-Pakri. Like what the second carbon which is there, like 5 carbons are there. I will show you here. So these 5 carbons are there. Okay, 1, 2. So this carbon maybe it will be below the plane. It will be what? It will be below the plane. So suppose this is the below the plane. This carbon is just below the plane. So it is called as a 2 prime C2 prime Indo-Pakri. Now, and what it is? Z is what? It is a mixture of the Pakri. It's not only having a single Pakri. It is a mixture type. So different books have different type of the like little bit of different values, but these are the authentic value which is taken from the reference books. One important thing, the BDNA is the right-handed structure and the ZDNA is left-handed. So it is a right-handed and this is a left-handed. Now, our point of discussion is that Watson and Crick given the they propose the DNA replication is a semi-conservative type of DNA replication. But whenever you will see the DNA replication cannot be done in this type of structure. Suppose this is our DNA. If it is a straight, then you can easily replicate it. But once it will be like a coil, how you will be able to replicate this one? Are you able to replicate this one? No. We have to make it in a straight way. Okay. So such kind of thing is done by the topology. That is the terminology which we are using. The topological change is very important to make it single standard and to make it possible to copy it. Okay. So this is something which we have to understand. For that, one scientist came. His name is Dilberg. So Dilberg came. He was the scientist who given the idea of the topological topoisomerase, basically the enzyme. So the making of the like unwinding of the DNA is very important. Without unwinding of the DNA, it is not possible to make a strain. So how it will be done? So Dilberg told we have to make a break in a DNA, then we will make a join. So break and joining is done by the enzyme which is called as a topoisomerase. Topoisomerase. So this topoisomerase is of two types. Type 1 topoisomerase and another is type 2 topoisomerase. Each having individual one type A and B type. But if you will see, basic difference in A and type 1 and type 2 is it will give a single strand break. What is it going to give? It is going to give you the single strand break, and another is what? It is giving double strand break. Double strand break. Now I am going to give you the one important question. So this topoisomerase is involved only in which phase of the cell cycle? Suppose, okay, in which phase of the cell cycle topoisomerase will be involved. So let me give you the S phase, G0 phase, G1 phase, M phase, okay, or one more option, all of this, or any of this, okay, any of this. Give me your answer. Please do the comment. So this class will be more interactive. Okay. Now, answer I will tell you now. Please pause and then give you the answer. So DNA, this topoisomerase is involved mostly in the DNA replication. We knowing that, but you must have know heterochromatin and euchromatin. So heterochromatin euchromatin concept was given in the transcription, and transcription will be occur everywhere, mostly in the G1 phase and the G0 phase, or even in the DNA damage repair and all this. So this topoisomerase is involved in the any of this cell cycle phase. Okay, it's basically I want to tell you the DNA, this topoisomerase is involved in the replication as well as transcription as well as damage repair as well. So it is involved in the most of the part. Now type 1 is giving you the single strand break, and type 2 is giving you the double strand break. So in type 1 there is a 1A is a one type and 1B is the another type. So 1B is the mostly popular enzyme in the DNA replication. In case of eukaryote, you will find the top 3 as the common DNA topoisomerase. Mostly topoisomerase are meant for the unwinding and relaxing of the DNA strand. But the enzyme which is called as a gyrase, where it will found? It will found in the prokaryote. This DNA gyrase, you must have know about it. This DNA gyrase is a positive supercoiling. It will induce positive supercoiling. But rest of the all the topoisomerase are giving negative supercoiling. These all are what? These are negative supercoiling. One more enzyme is there, DNA gyrase, and the reverse topoisomerase are involved in the positive supercoiling. Now 1 and B are the two type of the type 1. They are the two different type of the type 1 topoisomerase. So in one, what you will find, this is a DNA, and now let me draw with a two strand with a different color. It will be easy to understand. Okay, now. So suppose we made a one break here, and what we will do, we will pass this. It will pass from here, and finally you will get this type of opening. So what you found, there was a turn. Linking number is something in which we will understand. Linking number is what? Linking number is the number of the one strand passing the another strand. The number of times one strand is passed over another strand. Again I am repeating, the number of times one strand is passed over the another strand is called as a linking number. So what do you think, linking number is increase or decrease? So here 1, 2, 3. 3 times it is passing, but here only one time is passing. So the linking number is decrease. So by one number it is decreasing, linking number. Now it is unwinded just by passing of the one strand. In case of the 1B, what is happening? This is the DNA. This is the DNA. Now again you made a break here. But instead of passing of this blue strand, what it will be happening? This complete will turn on. This complete will be round. It will complete turn round, and it will form like this. It will form like this. One strand will be there. Now another strand came here, and you will find This will completely form making a round of a structure. Okay, so this complete round is done by the type 1b topoisomerase. Now this type 2 topoisomerase is giving the two double strand break, and it is a little bit complex. Suppose this is a such type of DNA is there. I'm making it more complex. So what you will do, you will make a cut over this, and then over this. So what you will find is all the DNA strand which is outward, it will go backward just by this. So you will find this complete DNA strand in a single strand just because of making of double strand, and then it is passing back to the position, and it is simplified into a single strand. Are you getting my point? Any doubt is there? At least you remember double strand will be break and then rejoin. Okay, and then rejoin. Okay, rejoin is important. Rejoin. Now these are the all things about the topoisomerase. Now let me tell you more about it. So type 1a is a more common topoisomerase in the prokaryote. And topoisomerase 3 is there in the eukaryote. It is in the eukaryote. Now you remember this one. You remember the main type of this. What are the main difference between this? And then you remember one more thing, that type 1, type 2 also having a subtypes. These are 2a and 2b. You remember this B is only in the archaea and plant. This B is only in the archaea and plant. This B is only in the archaea and plant. Please note in your notebook. Now we'll see Meselson and Stahl experiment, and then we'll wind this lecture. The Meselson and Stahl experiment was the fundamental experiment which is proving the Watson and Crick proposal. Okay, so Watson and Crick proposed that DNA replication is done semi-conservative way, but they are unable to prove it. Now the Watson and Crick, Meselson and Stahl, given the idea of the semi-conservative way of DNA replication. Semi-conservative way of DNA replication. So in beginning, what they thought, there can be this type of way DNA replication, semi-conservative way. Another, they thought there is a complete conservative replication, and one more type they thought, that is a dispersive replication. Okay, you can see this is a mixing of a DNA. So either it is a semi-conservative or conservative or it is a dispersive. So Dilberg's idea was his idea was good, the breaking and joining of a DNA, until the topoisomerase was discovered. He was unable to explain this semi-conservative way of replication. So his idea was supportive for the dispersive replication, but actually it is not happening. These two things are not happening. Only semi-conservative type of replication is happening, and it is done because of the heavy isotopes. Okay, so in nature we have a N14 as a natural. It is a natural nitrogen source. and its heavy isotope is what? N15 is heavy isotope. So N15 is a heavy isotope. In beginning, they took a DNA. They took a DNA and they replicated the E. coli in N15 medium, NH4Cl. Okay, so they put N15 medium and they replicated the DNA in N15. So all the strand now it is constituted with the N15. Now after that they transferred into the N14 medium. Okay, N14 medium, NH4Cl. This is the component. So they transferred into this, and then what they found? They found after DNA replication, like the timing of the E. coli replication is what? E. coli take almost 40 minutes for DNA replication. Okay, E. coli take how much time? It will take 40 minutes for the DNA replication. Actually, but it is E. coli is divided in 20 minutes. It is amazing now. It is taking DNA replication. Actually, if there is a one-time replication, it will take 40 minutes. But its cell is dividing in 20 minutes. How is it possible without the DNA replication? It is divided into 20 minutes. Again, I am saying without DNA replication, the cell division time is less than the DNA replication. How is it possible? Without DNA replication, any cell can divide. Let me know. Please do the comment. I will tell you the answer. Now, all the DNA will contain what? N14? No, it is not like that. So, after that, they centrifuge it. What they did? This is the first cycle. And then again they after this is the second cycle. Okay, this is the second cycle. In the second cycle, what is happening? Now the number of the copy of the DNA will be increasing. Okay, so this is also N14. Now I am making a DNA simply like this. They did a centrifuge. Okay. After centrifugation, they found two types of band. Okay, and they centrifuge into what? They centrifuge into cesium chloride. Why only cesium chloride, not in water? Anybody can tell us why it is a cesium chloride not in the water or not in another substrate? That is one most important conceptual thing which we will discuss in the end of this class. Okay. Now, so this two type of band we are getting, and this band is below because it having a N15 isotope. Okay, it is having what? N15 isotope, but also in the first type of the cycle, in this first cycle they got only one band, but in second cycle they got two band. In third cycle also they got two band only. Fourth cycle also they got two band. So they are concluded this heavy cycle, the heavy chain is consisting of N15 and N14. Are you getting my point? Are you getting my point? But the later, okay, make it as a one more cycle. You add third cycle. In the third cycle, you will get. In the third cycle, you will get also two band. In the second cycle also you will get two band. Now in the third and second cycle, The lighter band which is above, it consists of only N14, N14. So it is possible because of the semi-conservative. This N15 is coming from what? Parent. And this N14 is coming from the new strand. This two new N14 is also new strand. So they concluded if there is a DNA, if there is a DNA, let me draw here. So one strand is coming from the parent. This is my parent strand N15 and this is my new strand which is N14. So whenever the DNA replication will be occur, one strand is coming from the parent, another strand, the new strand is synthesized and it is half conserved, means it is semi-conserved from the parent. That's why it is called as a semi-conservative way of DNA replication. So even the first cycle was very important. In first cycle you will get one band only, the below band you will get in first cycle, but in second cycle, third cycle, fourth cycle, always you will get the two band. Now why they choose as a cesium chloride as a centrifugation material? This is the question. And how it is possible? These are the two questions. I need your interaction. Please do the comment and join our live classes which is given in the description box link. Now answer for this is what? Just before the ending of the DNA replication, it is a consecutive DNA replication happening. Okay. So before the ending of the one replication, the second replication is started. So let me show here. So this is the DNA replication is happening. Okay. Now until it will reach this point, the new DNA replication will be started here. Okay. Again then new DNA replication will be started here. So this by this way it will be able to complete the DNA replication within the time frame. Another question was that cesium chloride why we use. So usually in beginning the scientists use the ether, ethanol, other acetone as a centrifugation material. But the separation, the density was not that much, so they can differentiate N15 and N14. So the density is very important, which is provided by the only by the cesium chloride to differentiate N14 and N15. But in case of acetone, they are unable to differentiate, or in case of the water oxide, they are unable to differentiate the N14 and N15. That's why we choose cesium chloride for the density gradient. Please note it down. For the density gradient, they use as a cesium chloride. Hope so you enjoyed this video, and if you will have any doubt, please do the comment and please join our WhatsApp group, which is given. The link is given in the description box, and do subscribe and share this. Thanks for watching. We'll meet in another video. Jai Hind.